Welcome to our journey through the fascinating world of neuroscience methods. We begin by exploring the four core questions that clinical neuroscience seeks to answer. What is the brain made of? How does it work? How do we measure and manipulate it? And what can we infer from these measurements? Every method in neuroscience involves trade-offs between spatial precision, temporal precision, and invasiveness. Understanding these foundational questions sets the stage for all the techniques and innovations we'll discuss throughout this video. Let's break down the basic organization of the central nervous system, or CNS. The CNS consists of the brain and spinal cord, and it's divided into gray matter and white matter. Gray matter, made up of neuron cell bodies and dendrites, is where computation happens. White matter, on the other hand, contains myelinated axons that enable fast communication. The brain itself includes the cerebral cortex, which is responsible for higher cognition and voluntary action, as well as subcortical structures like the amygdala and hippocampus, which handle emotion, memory, and movement. The brainstem manages vital autonomic functions, such as breathing and heart rate. At the core of brain function are neurons, specialized cells that transmit information through electrochemical signals. Each neuron has dendrites to receive inputs, a cell body, or soma, to integrate those signals, and an axon to send outputs. The synapse is the junction where neurons communicate. At chemical synapses, neurotransmitters are released into the synaptic cleft and bind to receptors on the next neuron. These signals can be excitatory, like glutamate, or inhibitory, like GABA. This intricate communication network underpins all brain activity, from simple reflexes to complex thoughts. Neurons communicate using action potentials, brief electrical spikes that travel along the axon. At rest, a neuron's membrane potential is about 70 MV. When a stimulus pushes this potential past 55 MV, an all-or-nothing action potential fires. This mechanism prevents random noise from triggering signals and ensures information flows in one direction. It also allows reliable communication over long distances in the nervous system. However, most non-invasive brain imaging methods don't measure these action potentials directly, but rather the summed activity of many neurons. Human neuroscience, recording from single neurons, is rarely possible. Instead, we rely on methods that measure the activity of large groups of neurons. Neurons with similar functions tend to cluster together, so techniques like EEG and FMRI capture the summed activity of these populations. While this approach sacrifices some resolution, it allows us to infer brain function indirectly and non-invasively. Understanding these limitations is key to interpreting the results of brain imaging studies. Electroencephalography, or EEG, is a non-invasive method that records electrical activity from the scalp. What EEG actually measures are the synchronous postsynaptic potentials from cortical pyramidal neurons, not individual action potentials. EEG offers excellent temporal resolution, capturing changes in brain activity within milliseconds. It's direct, non-invasive, and relatively affordable. However, its spatial resolution is poor, and it's sensitive to artifacts like eye blinks or muscle movement. EEG is best suited for studying when processes occur in the brain, rather than precisely where. Ventrilated potentials, or ERPs, are derived by time-locking EEG signals to the onset of a stimulus and averaging across multiple trials. A classic example is the N170 component, a negative deflection about 170 milliseconds after seeing a stimulus, which is larger for faces than for other objects. This finding, first described by Benton and colleagues in 1996, shows that the N170 reflects early, face-sensitive perceptual processing. ERPs are powerful tools for studying category-specific brain responses and clinical conditions like prosopagnosia. Functional magnetic resonance imaging, or FMRI, measures brain activity indirectly through the bold signal, blood oxygen level-dependent signal. When neurons become active, they increase their metabolic demand, leading to changes in blood oxygenation. FMRI detects these hemodynamic changes, offering high spatial resolution and the ability to image deep brain structures. However, FMRI has poor temporal resolution and only provides correlational, not causal, information. It's best for identifying where in the brain activity occurs during different tasks. Transcranial magnetic stimulation, or TMS, uses magnetic pulses to induce electrical currents in the cortex. This technique can temporarily disrupt or excite targeted brain regions, creating a so-called virtual lesion. TMS allows researchers to make causal inferences about brain function, testing whether a specific region is necessary for a particular task. However, TMS is limited to superficial cortical areas, has coarse spatial precision, and its effects can spread through neural networks. It bridges the gap between correlational imaging and traditional lesion studies. Each neuroscience method has its own strengths and limitations. EEG excels at temporal resolution, but struggles with spatial localization. FMRI offers excellent spatial resolution, but is limited by slow temporal dynamics and correlational data. TMS stands out for enabling causal inference, but is restricted in anatomical reach. No single method is sufficient on its own. Strong conclusions require converging evidence from multiple techniques, each providing a unique window into brain function. Summarize this section. Neuroscience methods differ in what they measure and what they allow us to infer. This means that drawing strong conclusions about brain function requires converging evidence across different techniques. By combining methods, researchers can overcome individual limitations and build a more complete understanding of the brain. Now, let's shift our focus to the cutting edge, clinical neurofrontiers. Clinical neuroinnovation refers to emerging technologies and methods that move neuroscience beyond observation, toward direct interaction, intervention, and restoration of function. The main goals are to restore communication or movement, modify dysfunctional brain activity, and translate neuroscience discoveries into real-world clinical benefits. These advances also introduce new ethical, methodological, and clinical questions. Brain-computer interface, or BCI, is a system that enables direct communication between the brain and an external device, bypassing traditional motor pathways like muscles and peripheral nerves. BCIs are especially valuable for individuals with severe paralysis, locked-in syndrome, or spinal cord injury. While originally focused on movement and communication, BCI applications are expanding into cognitive and psychiatric domains, offering new hope for many patients. Most BCI systems follow a similar processing pipeline. First, they acquire neural signals, then preprocess the data to remove noise and artifacts. Next, they extract meaningful features from the signals and classify them to decode the user's intention or mental state. Finally, the control interface translates this output into action, such as moving a cursor or controlling a robotic arm. This structured approach is essential for reliable BCI performance. EEG is a common method for acquiring signals in BCI systems. It records electrical activity from the scalp, is non-invasive, portable, and relatively low in cost. EEG offers high temporal resolution, making it suitable for real-time applications. However, its spatial resolution is poor, and it's susceptible to noise from muscle activity or environmental sources. Despite these limitations, EEG remains a popular choice for many BCI applications. Higher precision, some BCIs use embedded electrodes, such as ECOG or intracortical microelectrodes. These methods provide both high spatial and temporal resolution and a strong signal-to-noise ratio. However, they are invasive, requiring surgical implantation, and carry risks like infection or tissue damage. The decision to use embedded electrodes involves weighing the benefits of precision against the risks of invasiveness. Exogenous BCIs rely on externally presented stimuli to elicit predictable brain responses. A common example is visually evoked potentials, where letters or symbols flash on a screen and the user focuses on their target. The brain's response identifies the intended choice. Exogenous BCIs require minimal training and offer high accuracy, but they are visually demanding and require intact vision and attention. Their flexibility is also limited by the need for external cues. Endogenous BCIs, in contrast, rely on internally generated brain signals, independent of external stimulation. For example, slow cortical potentials reflect shifts in cortical excitability, and users can learn to modulate these signals to control a cursor or select options. Endogenous BCIs offer greater autonomy and are not constrained by predefined stimuli, but they require long training periods and success rates can vary widely between individuals. Embedded electrodes come in two main types, ECOG, which are placed on the cortical surface, and intracortical electrodes, which are inserted directly into brain tissue. These methods allow direct recording of neural activity with high precision, providing access to fine-grained information. However, they involve surgical risks, potential infection, and long-term ethical considerations. These techniques blur the line between neuroscience and biomedical engineering. Neurofeedback is a technique that provides individuals with real-time feedback about their own brain activity, enabling them to learn voluntary self-regulation. The core principle is simple. If people can perceive their neural activity, they can learn to modify it. Neurofeedback allows researchers and clinicians to test brain-behavior relationships by directly altering neural activation, opening new avenues for therapy and self-improvement. FMRI-based neurofeedback offers high spatial precision and can target specific brain regions for training. However, it is limited by high cost and low temporal resolution, making it less practical for widespread use. Despite these challenges, FMRI neurofeedback has shown promise in helping individuals learn to control activity in regions like the amygdala, which can improve emotion regulation and potentially treat anxiety or trauma-related disorders. EEG-based neurofeedback is more portable and accessible than FMRI. It typically focuses on frequency bands or event-related activity, but offers lower anatomical specificity. Studies have shown that individuals can learn to modulate their brain activity using EEG feedback, leading to improvements in emotional and cognitive functioning. This approach is especially promising for therapeutic applications due to its convenience and affordability. Research by Keenan and colleagues in 2016 demonstrated that individuals could learn to downregulate amygdala activation through neurofeedback, resulting in improved emotion regulation. This finding highlights the therapeutic potential of neurofeedback for anxiety and trauma-related disorders. By enabling people to directly influence their brain activity, neurofeedback offers a new path for personalized mental health interventions. The rise of neurofrontier technologies, several ethical and clinical issues come to the forefront. These include the invasiveness and risks of certain procedures, the need for informed consent, concerns about long-term neural modification, data privacy, and equitable access to advanced treatments. Clinically, these innovations offer new rehabilitation pathways and hope for conditions once considered untreatable, but they also demand careful regulation and oversight. Wrap up this section. Neurofrontier technologies are shifting neuroscience from passive measurement toward active interaction with brain systems. This shift offers powerful clinical opportunities, but also brings significant ethical challenges. As these technologies continue to develop, ongoing dialogue and oversight will be essential to ensure their safe and equitable use. Recurring theme in neuroscience methods is the trade-off between spatial and temporal precision. Techniques like FMRI excel at pinpointing where activity occurs, while EEG shines in capturing when activity happens. Understanding these strengths and weaknesses helps researchers choose the right tool for their specific question and highlights why combining methods is so valuable. Another important consideration is invasiveness. Non-invasive methods like EEG and FMRI are safer and more widely applicable, but may lack the precision of invasive techniques like embedded electrodes. The choice between invasive and non-invasive approaches depends on the research or clinical goal, as well as ethical and practical factors. Most human brain imaging methods measure the activity of large populations of neurons, rather than single cells. This approach allows for non-invasive recording, but comes at the cost of resolution. By understanding how population-level signals relate to underlying neural processes, researchers can make meaningful inferences about brain function. Both EEG and FMRI capture the summed activity of many neurons. EEG reflects synchronous postsynaptic potentials, while fMRI detects changes in blood oxygenation related to neural activity. Although these signals are indirect, they provide valuable insights into brain function when interpreted carefully. EEG recordings are highly sensitive to artifacts, such as eye blinks, muscle movements, and electrical noise from the environment. Researchers use various preprocessing techniques to remove these artifacts and ensure that the recorded signals truly reflect brain activity. Proper artifact management is crucial for reliable EEG analysis. One of EEG's greatest strengths is its millisecond-level temporal resolution. This allows researchers to track rapid changes in brain activity, making EEG ideal for studying the timing of cognitive processes, such as perception, attention, and decision-making. fMRI stands out for its high spatial resolution, allowing researchers to localize brain activity to specific regions or even subregions. This makes fMRI a powerful tool for mapping functional networks and understanding the neural basis of complex behaviors. Unlike EEG and fMRI, which are primarily correlational, TMS enables researchers to make causal inferences about brain function. By temporarily disrupting or stimulating specific regions, TMS can reveal whether those areas are necessary for particular tasks or behaviors. No single neuroscience method is perfect. EEG struggles with spatial localization, fMRI is slow and correlational, and TMS is limited in reach and precision. Recognizing these limitations is essential for designing robust experiments and interpreting results accurately. To overcome individual limitations, researchers often combine multiple methods. For example, using EEG and fMRI together can provide both high temporal and spatial resolution. This multimodal approach leads to deeper insights and more reliable conclusions about brain function. One of the most exciting applications of BCIs is restoring communication for individuals with severe paralysis or locked-in syndrome. By decoding brain signals and translating them into text or speech, BCIs can give a voice to those who cannot speak or move, dramatically improving quality of life. BCIs are also being used to restore movement in people with spinal cord injuries. By connecting brain signals to robotic limbs or exoskeletons, these systems enable users to regain some degree of independence and mobility, opening new possibilities for rehabilitation. Beyond movement and communication, BCIs are being explored for cognitive and psychiatric applications. For example, they may help treat conditions like depression, ADHD, or anxiety by modulating brain activity directly. As technology advances, the range of BCI applications will continue to grow. The BCI processing pipeline involves several key steps: signal acquisition, preprocessing, feature extraction, classification, and control interface. Each step is critical for translating raw brain signals into meaningful actions, and advances in machine learning are making these systems more accurate and responsive. Developing effective BCIs involves overcoming technical, clinical, and ethical challenges. Technical hurdles include improving signal quality and reducing noise. Clinically, systems must be safe, reliable, and user-friendly. Ethically, issues like consent, privacy, and long-term effects must be addressed to ensure responsible innovation. Neurofeedback empowers individuals to self-regulate their brain activity. By providing real-time feedback, users can learn to enhance or suppress specific patterns, potentially improving mental health, attention, or performance. This approach is gaining traction in both clinical and wellness settings. The therapeutic potential of neurofeedback is being explored for a range of conditions, from anxiety and depression to ADHD and PTSD. By teaching individuals to control their own brain activity, neurofeedback offers a personalized, non-pharmacological approach to mental health care. As neurofrontier technologies advance, ethical oversight becomes increasingly important. Issues like data privacy, long-term neural modification, and equitable access must be carefully managed. Regulatory frameworks and ongoing dialogue between scientists, clinicians, and the public are essential for responsible progress. One of the most promising aspects of neurofrontier technologies is the potential for personalized interventions. By tailoring treatments to an individual's unique brain patterns, clinicians can offer more effective and targeted therapies, improving outcomes and patient satisfaction. Looking ahead, the future of neuroscience lies in integrating diverse methods, advancing technology, and addressing ethical challenges. As we continue to develop new tools and techniques, our understanding of the brain will deepen, leading to breakthroughs in both science and clinical care. The journey is just beginning, and the possibilities are vast.